friends, once you put away your warm-up, I need you to take out your Cornell note template. Yes, there are Post-it notes at your table. We're going to need them later. Let's keep them set down and not let them be a distraction while we take our Cornell notes today. On the screen, you see your EQ and your objective as always. We are not going to write this down word for word. What I want you to put in your EQ is identify, cell cycle, steps, DNA structure, replication. Identify, cell cycle, steps, DNA structure, replication.
Good morning, Pirates. Please stand for the Pledge of Allegiance.
While he's talking, you can go ahead and write down your objective for the sake of time.
Alex Chavarria and Leilani Silva, the Lady Pirates ran their way past the other contenders. The varsity boys finished a close second. Miguel Espinosa took second and Jose Segura finished fourth to continue to lead the Pirates. In the JV girl division, Abril de la Cruz won the thing, followed closely by Nelly Chavez, Anandeli Rodriguez, Mereda Marin, and Victoria Baez. Whipping the girls from Conlian, China Spring, among others. The JV boys took first place honors as well, with Noel Fajardo, Gia Armando.
All right, so hopefully you've gotten down your EQ and your objective at this point. We're going to move into our Cornell notes. Left-hand column, first heading, I want you to put DNA basics. There are some important scientists that we need to know a little bit of history about. I want you to know the name Watson and Crick. They proposed the structure of DNA.
Do I write that in the left-hand column too?
No, DNA basics goes in the left-hand column. Right-hand column is always the notes. Continuing in the right-hand column, I want you to put Franklin took an X-ray of DNA. Franklin took an X-ray of DNA. That was in about 1950s. And then Pauling told us more about the structure of DNA. So y'all can't see that on my screen, so I'm going to write. You can just put Pauling and structure. Franklin was the X-ray of DNA. Pauling is structure. Franklin is X-ray, Pauling is structure. Watson and Crick were the first to propose the structure. Out of those names, the ones you'll probably hear most often just in everyday life would be Watson and Crick. Anyone talks about DNA, they usually bring them up. All right, about to move on. We are still under DNA basics. Everything here is animated. So I want you to put in your right-hand column underneath your notes section, I know it looks like a heading, but we're going to include it in notes, that all activities of the cell are controlled by DNA. DNA is in the nucleus. Guys, what is the type of cell whose DNA would not be in the nucleus? Prokaryote, right? So in a prokaryote, where would the DNA be? Very good. So the prokaryote would have its DNA in the cytoplasm. You see up there, I abbreviated that. If you're not going to remember what that means, you can write it out. But I am the queen of lazy. And if there is a way to cut a corner and it still makes sense to me, I'm going to take it. There's another important academic vocabulary word here, and it's the word chromosome. Chromosome, guys, that's still DNA. It's just coiled up DNA. Have you ever taken, and girls, this probably makes more sense to you, a long strand of hair and twisted and twisted and twisted and twisted? What does it do? It folds in on itself, doesn't it? And it'll even pull closer to the scalp, right? That's basically what DNA does to become. It's what DNA does to become a coiled up chromosome. Now, DNA, its shape is actually a double helix. So when I say helix, and I want you to draw this in your notes, that just means like a ladder. So DNA is a double helix.
What's the bottom say?
DNA is a double helix. And as we move about this, you're going to learn that helical shape, that ladder of DNA, it's actually twisted. So it's like somebody took a ladder and twisted it.
Kind of like those spiral ones on those
Yes.
buildings in the air.
Yeah, you can think like a spiral staircase, sure. Give me a fist of 5 on this slide. How are we doing? Good. So let's talk about, left-hand column, we're going to change our heading now to what is the structure of DNA. And this, again, has all the parts. So left-hand column, your heading is what is the structure of DNA. Now, next heading, left-hand column, structure of DNA. Notice this drawing has lots of pieces. I only want you to draw one section. So we're going to draw a circle. That is our phosphate. Okay, throw it away. There's a sink right here. This phosphate is attached to a carbohydrate, which is one of our biomolecules. We call this is a 5-carbon sugar. So we got a phosphate, a sugar, and then we have a nucleotide, or a Actually, let me label these because it will make more sense to me. We're going to call this a nitrogenous base. Now, if you think back to biomolecules, we said clipin, carb, lipid, protein, and nucleic acid, right? And we said cho, chon, chomp. What we're looking at right here that I've drawn in black, that hopefully you've also drawn in your notes, what we're looking at here is a nucleic acid. I'm going to spin it, and you're going to realize we've seen. So the 4 bases are adenine, guanine, cytosine, and thymine. Give me a fist of 5 on this. How are we doing? Good? Okay. So the most important characteristic of DNA is these 4 bases. These 4 bases occur in complementary pairs. That's all I need you to write here. They are the alphabet of our genetic code, which controls activities of the cell. Our code occurs in complementary pairs. They are the alphabet of the genetic code, and they control the activity of the cell. So here in this drawing, this would be the outside of my ladder. Use this blue, it's wider. And these bases would be my rungs. And we'll talk more about that detail in a minute. But that's where I get that ladder helical structure, okay? So in your notes, you should say they occur in complementary pairs. They are the alphabet of the genetic code. And they control the activity of the cell. Excuse me. Fist to 5, how are we doing? All right, awesome. So, this DNA is still under the same heading. This deoxyribonucleic acid, let's write it down all the way out. That's its full name. That's what DNA stands for. Deoxyribonucleic acid. Write out the name. This whole thing of DNA is made up of those nucleotides that I showed you the structure of a second ago. Remember, it's my phosphate, my sugar, and my nitrogenous base. Those are my nucleotides. Deoxyribonucleic acid. Phosphate, the sugar, and the nitrogenous base. When I have all of this together, that equals a nucleotide.
So draw that out?
You should have already drawn that out somewhere. If you have it once, you're fine. I'm just reminding you of what I said earlier. Give me a fist to 5 on this. We good? Awesome. Oh, well, it's still there. It's just not highlighted.
What's the bottom sentence say?
The what?
The bottom.
That these things make up the nucleotides. All right. We're still talking about DNA. So I want you to know that DNA contains genetic information for cell function, growth, and division. We already said in eukaryotes, it's stored in the nucleus. Where is it in prokaryotes? It's in the cytoplasm. You have Post-it notes on your table. Take one Post-it note per person and look at this cell right here. It can either be a plant or an animal cell. On your Post-it note, I want you to write which one you think it is, and you're going to go stick it to either the either the blue or the yellow lab table. You've got 10 seconds to choose. We'll just write plant or animal. When I count to 0, when I get to 0, you need to get up, and you're going to stick it either where Miss Neighbors is standing or at the table behind Chantel. Am I clear? Chanel, I'm sorry. Chanel, I'm sorry. Here we go. 5, 4, pink and green, put your Post-its at the yellow lab. Yellow and orange, put your Post-its at the blue lab. I'll count down now. 5, 4, 3, 2, we should be there by 1. Quickly back to our seats. Okay, so now can you tell me, based on our learning from Unit 1 over biomolecules, DNA, if we were going to classify it as a biomolecule, Alex, which one is it?
Nucleic acid.
Yes, it's going to be our nucleic acid. So in our clipping, That's the last one. So it's got elements chomp, carbon, hydrogen, oxygen, nitrogen, and phosphorus, okay? So new heading, left-hand column of your notes. I want you to put now, we're breaking down DNA, put nucleotide. We've already addressed this, but I want to be sure that you fully understand. Left-hand column, put the word nucleotide. Left-hand column, write nucleotide. Right-hand column, I want you to put 3 parts. They are sugar, phosphate, and nitrogenous base. We know this, but I want to repetition, repetition, repetition. So I'm going to draw my sugar. He looks like that little house without a chimney. My phosphate is the little circle. My nitrogenous base is that rectangle. And when I put them together, I get... That's what it's going to look like all put together. So write your 3 parts, make your drawings. And I want you to know, deoxyribose and phosphate groups form the backbone of DNA. That, my friends, is important. Deoxyribose and phosphate groups form the backbone of DNA. One more time. Deoxyribose and phosphate groups form the backbone of DNA. We should have the 3 parts, we should have our drawing, and we should have named what's in the backbone of DNA. Give me a fist of 5 on this. How we doing? 2 minutes, I see, so we'll hang tight a second. So as we wait, can y'all tell me that cell we chose? If it was animal or plant, which one was it?
Plant.
Yeah, that was a plant cell. What I can look for to distinguish are the presence of a cell wall. So all cells will have membranes. The plant and the animal will have a cell membrane, but a plant cell will have a wall and an animal cell will not. Also, a plant cell will have a larger, more central vacuole. What other organelle will they have that animal cells don't? Chloroplast. Very good.
And
A bigger vacuole, yeah. All right, moving on. We are still under that last heading. You're not adding a new one. But I want you to make a subheading under the notes section that says nitrogen base pairs. These are the rungs of the ladder. They hold the strands together. And again, these are our 4 bases. My adenine, thymine, guanine, and cytosine. Now, these pair in a set way. They pair in a set way. Adenine will only bond to thiamine. I remember that by remembering apples and trees. So A with T. Apples and trees. I say A with T. I want you to say apples and trees. A with T.
Apples and trees.
One more time. A with T.
Apples.
Very good. Now the other set are cytosine and guanine. I remember that by remembering cars and garage. So I'm going to say, cytosine to guanine, and you're going to say cars and garage. Cytosine to guanine. Cytosine to guanine. So back to the top. Adenine and thymine.
apples and trees.
Cytosine and guanine.
Cars and garage.
Very good. So these bases will only pair this way. me a fist of 5. Are we good? Got a few people still writing, so we'll hang tight. Apples and trees and cars and garage. I'm about to wrap it up, so finish your thoughts. Get anything else you're lacking from a neighbor. Here we go. So the middle rungs, the base pairs, apples and trees, cars and garage, A with T and G with C. These are most directly associated with coding of genetic information. So base pairs, A with T and G with C, are directly associated with coding of genetic information. If it's highlighted, that's what you're going to write. You do not have to write every word. So I'm talking about the base pairs. That is my stuff here in the middle. So you see here, cars and garage, apples and trees, cars and garage, apples and trees. DNA, I know, is covered in 7th grade. So if you had a solid 7th grade science teacher, you should have talked about the structure of DNA at that point. If not, I'm sorry. We will cover it again. I'm not expecting you to remember it. Fist of 5, how are we doing? All right, moving on. So y'all see the sides? Those are the sides of the ladder. That's my phosphate, and that's my sugar. The rungs are down the middle. Those are my base pairs. Are you following me? All right. So again, my complementary pairs are, you don't have to write this again, but I really want to send it home. When I say C with G, you say C with G, A with T, C with G, A with T. So my C will always bond with my G. My A will always bond with my T when we are talking about DNA. So again, there's the sides of the ladder, and here are my rungs. So let's practice that. In your Cornell notes, I want you to write out this sequence. You're going to write out G, C, G, T, A, C, and you're going to draw a line. And I want you to write on the other side of that line what letter you think should go with it. I will show you what I mean. G is the first one. I know cars and garage, so here I should put a C. On your notes, you do the rest, and then we'll check it. What should be my second letter? Tell me. G. What should be my third letter? C. A. Very good. Here's another strand I want you to try. So in your notes, try this one again. Quietly on your own, and then we will check it together. I will start us. A goes with what? Very good. Continue for there. What should my second letter be?
C.
C. Third letter?
C.
Fourth letter?
A.
Next?
G.
Next?
T.
Next?
C. C.
Next?
A.
Very good. Y'all have done apples and trees and cars and garage. Give yourself a round of applause. Good job. All right. So, again, where in the cell, if it is a eukaryote, is the DNA located?
Nicholas.
What about if it's a prokaryote?
Cytoplasm.
Very good. So, new heading, left-hand column. I want you to put the cell cycle. Left-hand column, new heading, new ink. So change your pen color. Cell cycle. So life of cells is cycle. Beginning with creation, ending with division. Life of cells is cycle, beginning with creation, ending with division. So you should have started a new heading in the left-hand column, swapped your ink to a new ink.
You're writing down your notes.
So it's good to remember, however, not all cells are going to divide. For example, so not all cells divide. Cells of the central nervous system don't. So like, if you hear somebody talk about nerve damage, that's why. That's not something that if it gets damaged, it's going to grow back. Often when those are gone, they're just gone. But some cells will grow back. And what's interesting to know is what cells will and will not grow back often differ from organism to organism, right? Can lizards grow a new tail?
Yeah.
Can I grow a new leg?
No.
Maybe with enough stem cell research, but right now, today, if I cut my leg off, could I grow a new one? No, right? So it's different from animal to animal, and then it's even different in our own bodies from cell to cell, okay? Fist to 5 on this.
From scrape your elbow. Is your skin going back? Yes.
That's how. The cells are dividing and repairing. We're going to do one more slide and then we'll stop for the day. Flip to the back. Get another sheet. So we're still under cell cycle. I want you to know cells must replicate before they undergo mitosis. I know we have not used the word mitosis yet, but we will starting tomorrow. And I want you to know that all of that, the replication and everything, happens during what we call interphase. And I will go ahead and draw for you the stages of the cell cycle. We have G2, we have M, we have G1, and we have S. We start at G1, right, Miss Neighbors? I've gotten myself confused. G1 is first. Okay, so this is our cycle. This is where we begin, and we go this way. Okay? Over the next, tomorrow, we're going to break down each of these stages and talk about what happens in each of them. We are discussing cells. So we're talking about one cell in your body at any given moment, what is happening in it.
Functioning.
It's dying and growing back.
All right, here is what I want us to do. This is where we will stop today. Let's fold those Cornell notes and put them in the front pocket of your journal. We will pick up tomorrow. At that point, I would like all pens returned to the supply table, all journals put in journal boxes and placed on the shelf. Miss Neighbors, will you show our new friend how journal boxes work?